Okay, everyone, welcome back from lunch or whatever that hour-long break represented to you, wherever in the world you may be, for our online participants. And it is now my pleasure to introduce Dr. Cynthia Ryder from NIEHS, Division of Translational Toxicology, who will be talking to us about the Botanical Safety Consortium and how they have been evaluating NAMs for use in that area. Cynthia?
Thank you so much for the invitation, Nicole, to talk to you. So I'll be talking to you today about the Botanical Safety Consortium, as Nicole mentioned, and how we're evaluating NAMs for use with complex mixtures. So I was very grateful to the public commenter who set this up earlier in the day. So if you're not familiar with botanicals, they are a big business. I think about $12 billion of botanicals are sold in the US every year. And I love the Office of Dietary Supplements, their label database. You can search by product. So here we have botanicals that are on the market currently, and you can find over 31,000 products that are currently on the market. So if you think about this huge industry, so many botanicals on the market, really a very small fraction of those have comprehensive safety data available. So the National Toxicology Program has had a 30-year ongoing program in evaluating botanicals, and we've completed the ones on the left, and then we have several more studies ongoing. But it's just a small fraction, so we really need new approach methodologies to fill the gap. And I'm sure all of you could tell me much more about NAMs than I can tell you, but this is just to remind me to tell you that our definition of NAMs here is methods for assessing toxicity, hazard, and risk that don't include traditional mammalian bioassays. So we include in vitro assays, in silico models, as well as some whole animal assays like C. elegans and the developmental zebrafish. So as Warren mentioned in his talk, we all know the challenges that are inherent in using more simplified models of complex systems. But when we think about putting complex mixtures into those systems, we have to keep in mind that NAM testing platforms have been developed and optimized for use with single chemicals, like in drug discovery contexts or testing the active ingredients of pesticides. So when we think about putting complex mixtures in these systems, we have more challenges to overcome, like determining the concentration of the complex substances in the NAMs isn't always straightforward. We often don't know the active constituent. We can have multiple constituents that contribute to toxicity. It could be a very minor adulterant or constituent that could be the toxicity driver. We can have the potential for matrix interference, and this is increased with some complex mixtures. We can have some constituents that don't go into solution or that may precipitate in the testing media, and also in vitro to in vivo extrapolation is even more complex when you're thinking about these multiple constituents. So we all know the major efforts in NAM development, like Tox21 and ToxCast at EPA. And it's important to note here that these were mostly focused on single chemicals. There was some inclusion of defined mixtures where we know all of the constituents of the mixtures, but it really was focused on single chemicals. There have been some efforts to extend the application of these high throughput screening approaches to mixtures, and I'll just highlight two that I think are very relevant and interesting. The first is an EPA effort led by Barbara Wetmore, where they evaluated complex mixtures. They looked at 30 different fruit and vegetable extracts in a subset of the biomap assays. So here, these are 8 primary cell models that are stimulated to mimic human disease states like vascular inflammation and immune activation. And they found there were significant quantitative and qualitative differences between the extracts and the normal individual chemical ToxCast substances. So the fruit and veggie samples were 10 to 50-fold less potent than ToxCast chemicals on a molar basis. And at the maximum testing concentration of ToxCast chemicals, the fruit and veggie extracts displayed less activity. Another effort at the National Toxicology Program we essentially took our botanical library as well as some key constituents from those botanicals and evaluated them in a subset of TOX21 assays. So this included 20 endpoints like endocrine activity, nuclear receptor signaling, stress response, genotoxicity, and cell death. We found, unsurprisingly, that when we tested the individual constituents, they exhibited greater activity than the mixture. And overall, the distribution of activity was similar to that seen with TOX21 chemicals. But in both of these cases, we're really just putting complex mixtures into these established systems and asking, what do we see? We're not evaluating the systems for those complex mixtures and their performance. And that is where the Botanical Safety Consortium comes in. So this is a public-private partnership to improve botanical safety. And our mission is to evaluate the suitability of in silico tools and in vitro assays for testing these botanicals as complex mixtures. This is a visual of our Botanical Safety Consortium framework. It started out by identifying what are the priority toxicity endpoints that we're worried about with botanicals. So cardiotoxicity, genotoxicity, hepatotoxicity in particular. So we then formed technical working groups around each of these toxicities, and we asked them to do two major things. One, to identify candidate botanical ingredients that they thought would be positive and negative, and I'll talk more about that in the next slide. And then to identify recommended assays that really cover that toxicity. From there, we built a botanical library, and we've begun to evaluate that botanical library in the recommended test assays. And we hope to get to the place where we can recommend a framework and toolbox for evaluating these complex mixtures. So, sorry about the formatting on this slide. What we asked them to do in nominating botanicals was to pick botanicals that had really robust toxicity information available, from preclinical evidence in mammalian systems to human evidence from clinical trials or adverse event reporting, and if possible, in vitro mechanistic evidence as well. We wanted these to have known toxic constituents, to be available from reputable sources, which isn't always easy with botanicals, and to have robust analytical methods available. And in terms of identifying the assays, we really wanted them to have good biological coverage for all the important endpoints and processes for that toxicity. Of course, we wanted the assays to be reliable and reproducible. We wanted them to be sensitive, so minimize false negatives, be human relevant, and commercially available. So this is a snapshot of our botanical library, and if you're a dietary supplement enthusiast, you may notice that some of these botanicals aren't really in dietary supplements because they're poisonous. So Aristolochia, for example, has known genotoxic agents. Ephedra was taken off the market because of its cardiotoxicity, and oleander is a straight up poison. So those were some of our positives. And then ginseng and milk thistle are really pan-negative. So these were evaluated in 2-year bioassays, and they were among the cleanest substances that NTP has ever tested. So we also have a chemical analysis technical working group, and they've done a lot of work up front. This is a recent paper that was led by Saramya Waidanatha, the co-chair of the chemical analysis team, and it really describes the key steps that went into procuring the extracts or making them ourselves if needed, the evaluation of solubility in the vehicle control, characterization, trying to use authentic standards whenever possible, our work to quantify the constituents present, and analysis of contamination or adulteration. So one of the nice things about this consortium is all of our data will be publicly available on SEBS. So this is just the beginning where we've provided the chemical analysis of the different botanicals in the library. And then this is a snapshot of the assays that were nominated by each of the technical working groups. So for example, the genotoxicity working group recommended the Ames assay for mutagenicity and the in vitro micronucleus test, as well as the tox tracker. So the ones in green, we already have draft data available from the groups. The ones in black, we're still working on it. But just to give you an example of one particular assay from the neurotoxicity technical working group, they've been publishing on the side. This work was led by Remco Westerink at Utrecht University, looking at neuroactivity screening of the botanical library using microelectrode array recordings. So to give you an overview of how the assay works, basically they isolated primary cortical neurons from a rodent model. Then there was a 7-day exposure to each of the botanicals. They look for mitochondrial activity to evaluate cell viability, and then they use these electrode plates to record neuronal activity. So on the right side, you can see aconite, which you get the baseline first, and then you add the low dose of 5 micrograms per mil, and you see an increase in activity. And then at the bottom, we're just showing that there's a concentration response. With the high dose, you see greater activity. But the important point I wanted to make here is just these data aren't very simple. There's a lot of different things to consider. There's different directionality. So you can get an increase or excitation, you can get a decrease in activity or inhibition, or a biphasic response. And then with the strength of the response, it can change. Sometimes it's a minor change, or sometimes it's a strong change. So there's a lot to unpack with each assay. And when we look at the results from our botanical library, so the top in the peach color, those were recommended by the technical working group on neurotoxicity as positive for neurotoxicity. And you get different responses like hyperexcitation or biphasic response. And then we have our 2 pan negatives, ginseng and milk thistle. And then the rest were recommended by other technical working groups. So we're not sure what to expect with those. There's not a robust data available on what the predicted neurotoxicity is for those chemicals. So we do get a lot of activity with our positives and not as much with our negatives. That's good. But moving on, how do we integrate across the different assays that are recommended by the group? So here I want to talk about an example from our developmental and reproductive toxicity group and how we're starting to build these case studies around these different toxicities. So the DART group recommended 4 assays for inclusion in the program: the DevTox Quick Predict, the zebrafish developmental assay, a C. elegans assay, and an assay looking at gene expression in 4 different cell lines. And I'm just going to talk about the first 2 assays briefly and then give you an overall picture of how we're working through these data. So the DevTox Quick Predict, here we have induced pluripotent stem cells. We expose to the botanical extracts for 48 hours, and then we use LCMS to look at the ornithine to cysteine ratio. So you can see the graph on the right is what's supposed to happen if you have a teratogen. You get that decrease in the ornithine to cysteine ratio before you see any kind of cell death. And once it dips below that threshold that they've set, it indicates potential for teratogenicity. So here's an example. Ashwagandha was nominated by the group as a negative for DART effects. So there was good negative data for ashwagandha. And then blue cohosh, the bottom panel, was nominated for positive reproductive and developmental effects. So here we don't see any kind of decrease in the ratio with ashwagandha, and we do with blue cohosh, so that is positive. And then moving on to our zebrafish developmental assay, there are a lot of different endpoints here. So some of the endpoints are relevant to the neurotox group, some are relevant to the DART group. The ones relevant to the neurotox group, the photomotor response in both embryos and at the larval stage, and the larval startle response. But at the bottom, we have a bunch of different panels that indicate the different malformations you could record from the developmental exposure. And we have death at 24 hours and death at 5 days. So what we're looking for is developmental effects that occur without mortality. And this work was done by Robin Tanguay's lab at Oregon State University. So that's just describing some of the things. How do we go about putting them all together and making decisions about, did they perform well, did they not? And here I'm going to hugely caveat that the next slide is just very preliminary and we haven't made any decisions, and it's the beginning of the data analysis period. But just to show you what it looks like, the 2 botanicals that were selected as positive based on the entirety of data from animal studies and human exposures were blue cohosh and usnea lichen. And the 3 in blue were selected as negatives: ashwagandha, ginseng, and milk thistle. So if we look at the DevTox Quick Predict, what we see when we look at this data, so only one was out of sync. The ginseng was positive in the DevTox assay. So there we might delve into, is there some kind of ADME related explanation for the positive ginseng result? Is there a compound that doesn't get absorbed by the body that elicited a positive response in the DevTox Quick Predict assay? What you might notice about the next column is that absolutely every botanical was positive for C. elegans assay. So there we might say, okay, this assay is too sensitive as is when we just take the readout of any activity at all. So we might need to evaluate applying some kind of activity threshold that distinguishes between just normal bioactivity and an adverse event. Then with the zebrafish, so here I'm just taking the malformation data. So these were positive for eliciting a malformation without causing death in the animal. And what we see is there's a lot of mismatches. So both of the positives are negative, one of the negatives is positive. There, we might ask, is this endpoint or assay misaligned to the expected toxicity for some reason? And then finally, with our transcriptomic evaluation there, you might notice everything is blank. This is because additional work is needed here to distill this very complex data set into a simple yes or no call that could indicate potential for DART toxicity. So that's what we're working through right now. Our conclusions thus far are that it's important to keep in mind that NAMs were developed and refined for use with single chemicals, and that they really require careful evaluation for application to these complex mixtures. We think that botanical ingredients offer an amazing opportunity to compare NAM-based data to in vivo and human data. And we think this data can then be extrapolated to other complex mixtures like environmental mixtures, et cetera. The Botanical Safety Consortium is a public-private partnership that focuses on evaluating the performance of NAMs with complex botanical mixtures. And our initial results highlight the need to integrate across the different assays and better distinguish normal activity from adversity in these NAM platforms. And with that, I want to highlight that we're having our annual meeting October 10th and 11th in Durham with both in-person and virtual options. And there, each of the technical working groups will go into a lot more detail on what they've gathered so far and next steps. And with that, I will just acknowledge the people that were involved in the DTT Botanicals and Tox 21, and definitely the Botanical Safety Consortium steering team, especially HESI staff, Michelle Embry and Connie Mitchell and Julie, who make it all possible, and then the DART Technical Working Group for letting me steal their data and present it here. Thank you.
Wonderful. Thank you so much, Cynthia. Are there any questions in the room to begin with? Questions? I have a really quick question. And you know, we have the same home, so we can talk about this more later, but I am interested to know your opinions on sort of how to delineate between active and adverse, right? Because I think probably a lot of these compounds are they've been identified because of their potential neuroactive properties. Like that's why people take them, is because they have some sort of a stimulatory effect or some sort of an effect that may or may not be adverse depending on sort of the profile of the individual and their comorbidities and all kinds of other components. And so I wonder about sort of, I'm sure you all have thought about that.
Yeah, I mean, that's the whole key issue, is trying to make that distinction between activity, adaptive activity versus an adverse response. And so I think that's why we've chosen to focus on a range of botanicals that are, you know, really, really toxic, like drop dead toxic, and, you know, to try and distinguish what we're seeing with the entire literature database on what we expect to see versus what we see in these assays, to try and calibrate them using those positives and negatives.
And do any of the NAMs that you're using have any like genetic diversity incorporated in them? Any like multiple donors, or I don't think the zebrafish screens, they're just straight screens. You're not using any genetic manipulation. or
No,
anything
those
here?
were not. I mean, I want to say the hepatotox group might have some
Okay.
genetic diversity in there, but it's not. And one of the things that we were really concerned with was we're hoping that eventually the battery of assays can be used by people like Procter and Gamble and others. So we didn't want things that were so cost prohibitive or so nuanced that you can only use them in one academic lab, you know, under a certain condition. We wanted off the shelf assays that could be used.
Okay, thank you so much. Okay, now continuing with our colleagues from NIEHS Division of Translational Toxicology, I'd like to invite Eric Tokar to come up and talk to us about the novel tools and approaches group at ETT, and hopefully, here we go. Perfect.
Okay, thank you, Nicole, for the intro and for the opportunity to come up. It's my first in-person public forum and definitely the first time I've spoken at one. So excited to be here this afternoon. So as Nicole mentioned, I'm going to just give a brief overview of the Novel Tools and Approaches program within DTT. I'm not going to go into a lot of detail about it, but just to give you an idea of what we do. The advance. I missed something. Okay. And I'm going to breeze over the DTT organization. I think my other DTT colleagues have touched on a lot of this. So I will go into that. And then, like I said, overview of the NTA, and then get into some specific examples of some of the research projects that are in its portfolio, just to give you an idea of a small sample of the types of things, projects that are in its portfolio. So this is the DTT. There's 5 branches. I'm sure you all know, or you can find it online. And then a couple of my colleagues have already talked about the specific programs, the strategic areas of focus, exposure-based, health effects, innovations, responsive research, strengthening capabilities. NTA is in that bottom one, as you see, it's in the strengthening capabilities. And this just goes into a little bit more detail. NTA is different than almost all of the other programs, and that it's not one of the HEIs. Dr. Casey Warren talked about cancer HEI yesterday. There's also cardiovascular and DNT. There's exposure-based, like Cynthia just talked about with combined exposures and mixtures, and Will talked about yesterday with occupational inhalation exposures, responsive research. You see on the right, but as you can see, the NTA is down there at the bottom. We're in the strengthening capabilities. So we are really tasked with identifying or developing new and novel approaches, NAMs, you've heard that a million times, this forum, and hopefully these will help improve the research that's being done at the DTT through several different ways, but some listed there: increasing the testing throughput, increasing the speed of data acquisition, not taking as long to get the data that we need, increasing data accuracy, precision, more in-depth analysis such as modes of action or mechanisms. And that's where my group comes in quite a bit. We're in the mechanistic toxicology branch, so we like going into the weeds and looking, trying to tease out mechanisms, and also enhancing human relevance of the studies. So all of the research programs that I just showed in the that diagram, they have their research portfolios. NTA has approximately 70 projects in its portfolio, sort of broken into 2 different categories: bioassays and biological systems, and novel applications. Some examples of the biological systems: a lot of 3D. There's still some 2D as well, but 3D is becoming more and more prominent or popular, I guess, in these projects. Liver models, spheroids, a lot of several different organoid models. There's also some in vivo with the zebrafish and the 5-day rat assay. And the applications, those involve things like metabolomics, phenotype screening, transcriptomics, data sequencing. And of course, there's some overlap with these. We used a lot of these applications for a lot of the models that we're developing or identifying. So that's it. That's what the NTA is and what we do. So I'm going to jump into some of a very small percentage of the 70 or so projects that are in there. And all of these are going to be pluripotent stem cell based. And that's mostly because that's ones that my group is in charge of. So I'll be able to hopefully speak intelligently about them and have some idea of what they do and what they're there for. So a few years ago, my group switched focus a little bit, started looking at developmental toxicology. And so we wanted to look at very early life development. So we thought the pluripotent stem cells, and this includes embryonic stem cells and iPS cells, wanted to look at these because we didn't think this is sort of a relatively understudied area. There didn't seem to be a lot of really good in vitro models. There's a few in vivo models that were kind of looking at this, but you all know the common issues with in vivo, labor intensive, a lot of money, a lot of time. So these pluripotent stem cells sort of help us get around those things, and they're physiologically relevant. Everything we use is human. They're rapid, especially compared to the in vivo, gets us to that high throughput format, mimics those very early events in development, embryogenesis, organogenesis. And using these models, we can study very defined stages. And this was more interest from my group. I've always been interested in trying to tease out windows of susceptibility, and these models give us opportunity to do that. I'll show an example in the slides. And of course, what we're all trying to do, use fewer animals in the research. So we have developed several, we call them model systems, and each of the systems has these 3 different components showed here. And each sort of component is a kind of a model in itself. So we refer to them as our model system. This is the cardiac model shown here. We also have neuro, renal, hepatic, and colon. And they all follow this, they all have these same 3 components. And everything starts in that little gray box, might be a little hard to see, but that's a colony of pluripotent stem cells, embryonic stem cells in this case. And from there, we can go to the embryoid body formation. We could do your standard monolayer 2D directed differentiation to theoretically any cell type that we have a protocol for. In this case, we're pushing them to cardiomyocytes, which we have a GFP reporter in for the NKX 2.5 gene, which is a marker for cardiomyocytes. So we can just monitor that very easily. And just when they turn green, we've got cardiomyocytes. And then the third component is the 3D at the bottom. I don't know if you'll be able to see, this is cardiac again. You might be able to see it beating in there, sort of mimicking the beating heart. So all of the model systems have this same format. We can collect the same types of samples, analyze them the same way. Everything's the same. The only difference is the number of days for the differentiation. Cardiac in this case is about 8 to 10 days. Neural is about 40 days. And then of course the differentiation kits we use. But we can test the same thing, same chemicals or same mixtures, whatever we want. We can do the same. It works the same for all of these different 4 or 5 different systems that we have. So now I'll get into some more, little bit detail on some specific projects. The first one was the embryoid body model. We're using this mostly for screening teratogens. And that's because the embryo bodies or EBs are, okay, it's the wrong slide deck, but okay. They're just free floating aggregates of pluripotent stem cells, and under the right conditions, they will spontaneously differentiate and start forming cells from each of the 3 germ layers. So that allows us, they're good for studying teratogens. And we've worked it out where we can get a single embryoid body in each well of a 384 well plate. So it gets us to that high throughput format. And then once we have them after 8 to 10 days with or without chemical exposure, we can do same type of things you do with any other cells, collect RNA for PCR, transcriptomics for sequencing. We can do immunofluorescence, immunostaining for protein expression, co-expression, localization of cell types within the EB. We can also, throw in some metal conjugated antibodies, and then run them through our Helios system for CyTOF, and that will help with proteomics. And once we have all of those things, we sort of just plug and chug. We plug all these data into our artificial intelligence system that Ian, as a postdoc that was working on this project, plug them into this AI system, and with the machine learning and deep learning, which I have no idea what it does, but it pretty accurately, between 80 and 90% accurate with the predicting whether a chemical is going to be a teratogen or not. So the next project, one of them using the cardiac model system, we always say we're looking at early life development, in this case in the presence of toxicants or chemicals. The toxicant in this case is cadmium, and that's because my group has a ton of experience with cadmium when we did all our cancer modeling, and also because maternal exposure to cadmium is a pretty high risk factor for congenital heart disease in the offspring. So you can see, using the GFP reporter we have, we can just see how the increasing concentrations of cadmium inhibit the differentiation to cardiomyocytes. And then looking at the bar graph on the bottom, and breaking this into the windows of susceptibility I mentioned. Two different windows, the mesoderm induction, which is from day 0 to 2, and then cardiac induction from day 2 to 4. With cadmium, if we treat only during the mesoderm induction and then remove cadmium, then you get the significant inhibition of cardiomyocyte formation, which is in the black bars. But if you skip that stage and start the cadmium at the cardiac induction phase, so day 2 to 4, and then throughout the process, you don't see any inhibition of that differentiation. So this gets us to those windows, as I mentioned. And interestingly, when we did arsenic the exact same way with this, another inorganic, another metal, you see very, very similar percent inhibition of cardiomyocyte formation, but it had the opposite window of susceptibility. Didn't do anything when we treated only during mesoderm, but when you started it at the cardiac induction, that's when it had its effect. So I think a good example of how we can find these windows of susceptibility with some of these models. So when we did this and looked at genes and transcription factor signaling pathways, of course, these were all highly downregulated with cadmium. All these genes and transcription factors associated with cardiomyocytes and cardiac function. So we wanted, as I mentioned, we're the mechanistic toxicology branch. So we wanted to dig into what mechanism could possibly be causing this. And it turns out it was an epigenetic mechanism, specifically histone trimethylation of the H3K27, lysine 27. When we added a specific inhibitor of this methylation, it pretty much completely removed cadmium's ability to inhibit that differentiation. So even in the presence of cadmium with this inhibitor, there was no inhibition that you saw without that inhibitor. So epigenetic mechanism, specifically dealing with histone methylation in this case. So again, just an example of the types of things we can do with these models and all the mechanistic stuff we're trying to get at in our branch. So that was all the 2D stuff. Looking at 3D, the immunofluorescence here just is characterizing the cardiac organoid. That's just 3 or 4 of the cells. In these, we've seen cardiomyocyte, we get cardiomyocytes, cardiac fibroblasts, endocardial cells, endothelial cells, atrial and ventricular cardiomyocytes in these organoids. So we feel they're very good organoid models for studying cardiac development. But what I really want to point out with this is more of a functional test we are doing. And you see in the bar graph in the middle, that's the frequency of the beating that I showed in that movie and one of the earlier slides of the video. And you can see as low as 1.0 micromolar cadmium, which is certainly environmentally relevant. You see about a 50% inhibition of the frequency of the beating in the compared to control. And then the heat map there just shows a bunch of genes and transcription factors associated with cardiac development and function. Top row is the day 8 control organoid. Middle is the day 8 cadmium organoid. And you can see the pretty significant effects cadmium has on all of those genes and transcription factors. Um, so trying not to go over, sorry. So another project, again, using with the same PSC based models, this one I'll talk about is our neuro model. This is with cell-free DNA, looking at these in these systems. So I'm sure you've all heard of cell-free DNA. It's been around, been known about for 80 years or so. There's a lot of recent interest in the past 10, 15 years, especially in the cancer area. But it's been used for a while for things prenatal screening, fetal gender determination, that sort of stuff. But there's a very limited understanding, or really anything out there. The cfDNA changes during organogenesis and differentiation. And Brian is the postdoc working on this project. She and I were just talking last week. And when we first started this project about 2 to 3 years ago, we could find, I think, 2 papers in the literature dealing with cfDNA and organogenesis. And now she was telling me last week she can find 6, and 3 of them are ours. So there's still not much out there. So we're still learning a lot as we go with this. It's still early on. But basically, we just have our organoid cultures. We collect the cfDNA from the conditioned medium and then spin it down, use some kits to isolate it. And then we can do the downstream analyses, sequence identification, all that. And the goal for the end, I guess, is to hopefully find some targeted biomarkers that we can use for, chemical effects or disease formation or whatever. We're not there yet. We're still kind of early, but developing this model, obviously, like I said, there was nothing out there. We didn't know what we were going to get from it. Turns out we can get cfDNA from these models. It's pretty low concentrations. We can do our fragment analysis. The majority of the fragments are between 100 and 200 base pairs, but right around 150, 160 is the most common size. We can get genes of mitochondrial and nuclear origin that we measure with droplet digital PCR. So we can get these. There's enough there. We can do quite a bit of downstream analysis. So we're hopeful we'll be able to start with that biomarker discovery soon, but we have done a little bit of work with this. We've done some whole genome sequencing of these fragments to characterize cfDNA. And when we enter the genes that intersect with those fragments into gene ontology and disease ontology, this is what comes up. Maybe not surprising, but a lot of terms involved with neurodevelopment and brain structure and organization. Disease ontology was interesting. I thought the terms that popped up were intellectual disability and autism. So this, excuse me, tells us that the cfDNA we're getting from these is, of course, relevant to brain tissue, human neurological disorders, and development. So again, hopefully looking more into this, we'll be able to do some biomarker discovery. And Brian, she's the only postdoc I've got right now. She's got about 8 more months on her contract. Hopefully she'll stay there the whole time. And she's talking about doing a lot in the, hopefully very near future, comparing what we get from these models to human samples. We'll start exposing these organoid models, not just the neuro, but we've done some work with the cardiac model as well. So chemical or toxic exposed organoids, see how that changes the cfDNA. And then we might try to do some patient-derived iPS lines. You get the cells from a patient with a known neurological disorder, in this case, reprogram it, and then differentiate it to the neural organoid or the cardiac organoid. And then you can collect the cfDNA or just do genetic analysis, whatever. They do it a lot with cancer. You recapitulate tumor formation. It's an idea we've got for this. And then finally, this will be really quick. I only have one slide. We very recently, this one is almost ready for prime time. We just got 1 or 2 more things to look at to characterize these, but we have a dual reporter system in the human embryonic stem cell line. We use CRISPR gene editing to insert markers for nestin, the GFP, which marks for neural stem cells, and then N-scarlet for tyrosine hydroxylase, which marks for DA, dopaminergic neurons. As you can imagine, this let us very easily and rapidly image and measure the different stages of differentiation and neuron development in this case, and monitor the formation. And then, of course, analyze them however we want. Genetic analysis, molecular analysis, all that stuff. But like I said, I just ordered the last antibody we need last week. So hopefully it comes in this week and we'll get that done soon. And this paper will be out the door soon. Another paper, but this will be out the door. So to summarize real quick, I think I've shown several useful model systems for improving the science, DTT. Try to address all the different tasks assigned to the NTA, throughput, rapid data acquisition, and so on. And I think the models offer a great opportunity for studying the effects of early exposures on later life disease. And this is a very small percentage of the 70 projects. They're not all pluripotent stem cell based. There's a pretty wide variety. And I think they're all really helping with the science in DTT. And then finally, I need to thank everyone on this slide, the current members of the NTA program listed on the left, members of my group, current and former members who recently left and did a lot of the work that I showed you today. And that's it.
Thank you very much, Eric. Do we have any questions in the room? I have a quick question. So how does the NTA prioritize which methods and technologies and tools you look at?
So I just joined the NTA like less than 2
Plausible
months ago. So
viability.
yeah, I'm gonna I'll punt that to them and throw them under the bus, but I honestly don't know.
Well, we have a coordination meeting between NYCEDM and the NTA on the calendar, I think
Sure
next
you
month.
will.
So no, I'm excited for that. Thank you so much. Okay.
There's a.
Oh, sure. Randy, you can go ahead and unmute if you want to ask your question.
Yeah, I was going to ask a very similar question, like for cardiac, neural, there are, commercial entities out there with very nice platforms for screening and stuff. How do you guys determine whether you're going to do something in-house or just collaborate with some of these commercial entities who have, very reproducible platforms already?
What, can you repeat? I couldn't hear. They were.
It's a similar question, but also including consideration of commercial entities that have reproducible platforms. How do you determine whether you'll collaborate with them?
I just joined the NTA months ago. I don't. A lot of the other projects do use the commercial stuff. These were just projects that my group was asked to try to develop. So we do it in-house, I guess.
Thanks, Eric.
Okay, so now our next presenter is our colleague, Dr. Jesse Carter from the U.S. Department of Agriculture, AWIC, the Animal Welfare Information Center. And so let me find your slides and share screen. Okay, great.
Awesome. Thanks, Nicole. I'm always thrilled when I'm invited back to speak at this forum because I feel like my presentation is a lot less science and research based than what else you've heard today. But, I'm thrilled to be part of ICFAM and attend these forums and, learn about all the cutting edge research. So like Nicole said, I'm from the USDA's Animal Welfare Information Center, and I'm going to talk about who we are, what we do, and kind of how we support information around NANS and toxicology. Oops, sorry about the formatting. So to start out, I want to give a little bit of context of where AWIC is located, or Animal Welfare Information Center. And so we are located at the National Agriculture Library in Beltsville, Maryland. So, pretty close to here, the big, tall brown building by IKEA. Really the main mission of the Agriculture Library is to serve as the, information resource of the agriculture community. And we don't only just serve USDA or federal stakeholders, but we do serve the public and private sector as well. And so within NAL, there are 8 information centers within the customer services branch, and one of them being AWIC. So I just want to provide a little context there. And then so with a little history about AWIC is that we were created in the 1985 amendment to the Animal Welfare Act. And really the intent was to serve as an information service to support those involved in animal research, specifically to help provide information on employee training or those involved in animal research. And then also provide information that could prevent unintended duplication of animal experimentation as determined by the needs of the research facility, and on improved methods of animal experimentation, which could reduce or replace animal use and minimize pain and distress to animals. So really our stakeholders are, the animal research community. So attending veterinarians, institutional animal care and use committees, PIs, animal care staff, really anyone involved in animal research and looking for literature and information on NAMs and animal use alternatives. And so even further, the Animal Welfare Act put out regulations that says that, employees in animal research have to receive training in the 5 following areas on the slide. And specifically, number 5 talks about utilizing the services at the National Agricultural Library, likely implying AWIC, because we provide information on, appropriate methods of animal care and use, animal use alternatives, prevention of unnecessary duplication of research, and really the intent and the regulation of the Animal Welfare Act. And so now that I've gone over a little bit about our history and kind of who we are, I wanted to go over some of our products and services that we offer. So of course, I wanted to start out with our website, which is our main information source and where you can find information on NAMs and animal use alternatives. More specifically, we provide bibliographies on a variety of alternative research topics. We also have information on mandatory and regulatory testing guidelines. We, of course, cover the Animal Welfare Act and really any other animal welfare laws. And in general, how to conduct these literature searches to find this really technical and scientific information. And of course, if you have any questions or can't find something you're looking for, we're always just an email away. And I'm not going to take you through all of our web pages, but I did want to point out this one because I think it's relevant to our audience. So this is NAMs or 3 Rs and ecotoxicity testing webpage. And this actually came out of a request from the ecotox work group within ICVAM back in 2020. And so we built out this page and included one of our main products on this page is our bibliography on new approach methodologies in toxicology. And really that's the small team of AWIC kind of doing these very comprehensive, almost systematic review literature searches on this topic and hand selecting citations to go into this bibliography, such as, in chemico, in silico, in vitro methods. And we do update it every year, actually around this time. But the one online now is from 2010 to 2023. And then one of our other sections, that's where it is, that I wanted to highlight on this page is some additional literature, but these are set up in more of what we call our canned literature searches. And so what this means is that we've predetermined some search strings of different databases. And so if you click on see all results, it's going to take you to the most updated literature on, this topic, NAMs and ecotox. And so one advantage to this over a bibliography is it's going to always provide the most up-to-date literature, but on the downside, there's likely going to be irrelevant citations in those results that you're going to have to sift through. Whereas the bibliography, again, we went through every citation and hand selected them. And then we also have an extensive webpage that is highly used on literature searching because it's not always, just typing in a few keywords into Google Scholar, and there is a much more systematic and robust approach to this. So we provide step-by-step instructions on how to do this for NAMs. Some other things we provide are search examples that you can mimic, cheat sheets and worksheets, and then information on how to, plan and kind of publish your guidelines in the literature. One initiative I wanted to talk about us working on is, again, we're a library information source. And so overlapping with library science and animal use alternatives is we're developing what's called literature search hedges or search filters. And so what that really means is they're predetermined or pre-developed literature searches that essentially anyone can come and copy and paste and put into PubMed or Web of Science or wherever and find information on a specific topic. So the real advantage to this is it takes away, again, the library science from non-library folks, brainstorming keywords, knowing what search syntax to use, what terms to tag it with, and just, you can copy and paste and put it in the database and find what you need to find. And so what we've done is focus this on animal use alternatives. So some of the ones we published so far are on alternatives to painful and distressful procedures in animal research, like alternatives to oral gavage in rodents. We also have a number on search hedges on social housing of lab animals, and then some general lab animal hedges that you can copy and paste and use. And again, we're still working on developing a lot more for NAMs, which I'll go over on the next slide, as well as other general animal research and animal welfare. So we're hoping to publish more of those later in the year. The specific one I want to talk to you all about, though, is our collaboration with AltBib. And which, for those of you who don't know, AltBib is essentially a PubMed search filter that retrieves citations on alternatives to animal testing. And it's actually hosted on the National Toxicology Programs website. And so what we've done is work with some toxicologists or subject matter experts at NIEHS to develop search hedges specifically for new approach methodologies in toxicology. Specifically, right now, we're working on the ones that you see on the slide. And so again, what this is going to do is allow you to find, the most updated literature on these topics with more than just a few keywords. So it's a really robust search. And so we've gone back and forth and already drafted multiple versions of each of these fives. We've tested and gotten feedback from the SMEs because, of course, we're not toxicologists at AWIC, but we do a lot of these searches. So it kind of gives us a starting point. But we're actually excited because we think they should go live on AltBib's website in the next couple of weeks, or really next couple of days. So we hope to develop more in the future. And I just wanted to show you an example of what a search hedge might look like, just to give you an idea of how they're organized. And so keep in mind, though, this is a much redacted version of what actually a search hedge looks like. Usually they're pages long because, again, the point is to be as comprehensive as possible. But for brevity, to not overwhelm you, and mostly to fit on the slide, I've drastically cut those down. So the terms in blue all have to do with acute inhalation exposure and, or pulmonary toxicity you'll see as a keyword, or just inhalation toxicity. So that's part of the hedge for acute inhalation exposure. And then it's combined with the terms in green, which are all related to NAMs or animal use alternatives. So this hedge is specifically built for PubMed. And so if you were to copy and paste this in PubMed, what it's telling the database to do is to retrieve all citations related to NAMs in, acute inhalation exposure toxicity. So, and then on the right-hand side, you can see some of the selected citations that it's going to bring up, all very relevant for, on microphysiological systems like organoids or lung on a chips that are focused on respiratory disease or development. And I mentioned this a minute ago, but this is the AltBib website. And so you can just go to this link on the slide, and hopefully again in the next couple of weeks, you will be able to see those 5 NAM topics I showed you a couple slides ago underneath the section of modified PubMed search strategy. Those are the current topics that have been up there forever. And hopefully we're going to add those 5 very soon. And then in the future, we're hoping to still work with AltBib and NIEHS to update the ones that are already up there now, as well as, create new search hedges on this topic to overall just provide a more comprehensive list of this type of research and make it more visible to the researchers and really the public. Okay, and so now into some of our more established services that we always offer. So one of them, and some of you may be familiar with this, is our live workshop on meeting the requirements of the Animal Welfare Act. So this is free, it's virtual. We offer it 3 times a year. Our next one's in October. And really what we discuss in this is, a variety of topics, some being the Animal Welfare Act and its requirements specifically related to animal research. We go over examples and define NAMs and 3 Rs. But one of the big components is coming out with a, improved understanding of how to conduct these very, systematic kind of searches. And again, it's free, and it's just a great way to kind of learn this information for anyone really in the research or animal research community. But if you can't make our live workshop, this is always an alternative if you want to still have access to the information. It's free, it's self-paced, it's pre-recorded. It's pretty much the same information. Again, you just kind of go through modules and take it on your own time. Again, a great resource, but it's not going to have that live interaction and discussion that our workshops will have. And then the last training opportunity that we offer is our customizable training. So like the name implies, they're completely customizable and free based on request. So we can cover any of the topics on the slide, as well as a couple other topics, but those are our main ones. And we can do it to any institution or agency who requests. It's free. We can do in-person ones if they're local, or of course virtual. And again, the length, the time, the date can all be, figured out at a later date. We also have a literature searching assistance service. So basically what this is if a PI is having issues finding NAMs or animal use alternatives for a specific research topic or even a procedure or animal use protocol, they can reach out to us and we will conduct the search for them. One reason we started this service, and it's gotten a little bit expanded since the beginning, is because when PIs conduct animal research and there's going to be painful procedures involved, they have to consider alternatives. And usually the main way PIs would consider alternatives is through a literature search. And so that's required. So we do see a lot of PIs using our service, but we can also do it for other purposes as well. And then lastly, our newsletter. If you're not already signed up and you're interested, feel free to subscribe. It's very short. We only send it out once a month and we just cover a lot of the information that I went over, like NAMs and literature searching, updates with the Animal Welfare Act, et cetera. And with that, I'll put up our contact information and, happy to take any questions.
Great. Thank you so much, Jesse. And while Dana's going to the mic, I will just give a quick shout out to Kathy Sprinkle from the NICEDM group, who has been absolutely instrumental in working with Jesse to both develop those hedges and to get them accessible online. And that since Outbib was transferred to NICEDM a couple of years ago. I think,
in 2020.
Thanks very much. Dana Connors, Foundation for the NIH. Potentially totally irrelevant, but is AWIC specific to 3Rs and tox testing, or do you also provide resources to the agriculture community for other animal related issues like antibiotic use, breeding, housing, that kind of thing? Can you talk a little bit about that?
Yeah, great question. We serve every animal research community. We get a lot on agriculture, especially alternatives to antibiotic resistance. We do a lot of literature searches on that. We actually have a couple other information centers that can help find literature on that topic. We do a lot in toxicology because we know there's so much animal use. And so that's kind of why we tier towards that. But we do a lot of biomedical, really anything that falls under animal research and pain and distress we cover. Yeah. Thanks.
Okay, wait, literally.
Give him a second.
All right. Jesse, could you talk about the types of data that you are now accepting alternative data for, thinking about veterinary vaccines and the non-animal testing methods that may be available?
Thinking about the
Vaccines.
Oh, yeah. That's not really up our alley at AWIC as part of the library. We're really just an information resource and service. That's going to be more so with APHIS, the Animal Plant Health Inspection Service, which AWIC works really closely with, but they're the regulating body and kind of the ones organizing and dealing with that data. We're more the providing information on the intent and use of the Animal Welfare Act regulations. So sorry, but.
Okay, great. Thank you so much. Okay, so we are now scheduled for a break. We are now ready to reconvene. Rebecca is here. Okay, so our next session, we will hear a couple of presentations by our colleagues from the U.S. Department of Defense. And the first one will be from Rebecca Kluhl. And go.
Hi, I'm Rebecca Kluhl. I am a contractor for the U.S. Air Force. I'm embedded at the Air Force Research Lab, and I am the team lead for the predictive risk team. within the Forest Health Protection Program. So that was a lot of words, but we're really focused on preventative medicine to protect against chemical, biological, and physical stressors that could reduce performance, cause injury, or reduce mission success. I don't know how to get to the next slide. Oh, sorry. Okay, got it. Okay. Yeah, so standard disclaimer, I do not speak for the United States Air Force, everything in here, or the United States Space Force either. I am here as giving you my opinion as one of their employees. And we didn't steal any images from anybody. Okay, so for operational chemical risk assessment, I don't need to tell you all about chemical risk assessment, but I want to just highlight a couple of the pieces of the pie here that we're focused on. We need to make really quick decisions sometimes for what the potential risk might be, both in garrison and in deployment and in emergency situations and in wartime. So we have some kind of additional factors we need to consider when we're doing a chemical risk assessment. And a lot of these factors, you'll hear this probably from Dr. Adams after me, but a lot of these factors we need to consider are also true for like first responders, right? So there's of course the ingestion or dermal contact or inhalation exposure routes. And there's individual susceptibility factors and what these chemicals are going to do in the body. But in addition to that, we have all these physiological stressors in extreme environments we need to consider. So you might be deployed to the Arctic, you might be deployed to the Middle East, you might be in the space station, or you might be really deep underwater. Air Force doesn't do deep underwater, but other military branches do, as far as I know. But anyway, so we do need to think about these things. There might be exercise-induced fatigue, there might be altitude or G-forces, and so these can affect the concentration at which the chemicals are going to affect your performance. And of course, short effects on performance can have catastrophic effects in certain circumstances, right? So we're trying to think of all of those things and bring them together into a comprehensive risk assessment. And when I was brought into this team, the challenge I was given was, let's develop a NAM-based risk assessment approach that does not use animals as much as possible and is able to, or does support really rapid decision-making, even in the field decision-making, where maybe it's an austere environment where we don't have comms. And so we've been thinking, how do we bring all of these tools and ideas together that we've all been talking about for the last couple of decades and actually start making decisions? And as we make those decisions, or like as we do these projects, let's test these tools out and see how well do they do in the real world. Let's get some, everyone's doing case studies. We're also doing case studies, but more focused on the Air Force in particular. So of course, in a modernized tiers risk assessment, you start with in vitro modeling and maybe move into high throughput screening and then more organotypic models. These have different purposes, you all know, and allow you to do different things like prioritization or quantitative risk assessment. Because my team is the predictive risk team, we're very focused on tier 0, or the idea of building predictive models that will avoid the need for testing altogether, or at maximum high throughput testing, where we don't have to spend a lot of time and a lot of money before we have a go, no go decision. That's my Air Force speak for you. Go, no go. Okay. Okay, so we are a product development component. So our money is not basic research, not even applied. We're really product development. And so we are focused on building products that can be used in the near future and also testing these products to ensure that they actually are doing what they need to do. At the sort of base of what we do is a large research project called the Predictive Risk Capability Build. It's exactly what it says on the tin. We're building the capability. And so we're bringing together databases, we're building risk prediction models, or taking risk prediction models that already exist and applying them to the Air Force. If they already exist, we don't want to make them again, right? We want to grab them and make them more useful to us. Exposure models where we can, I'll show you some examples. And then we're bringing all these together into our toolbox. This is an acronym because the Air Force likes them. It's the Chemical Exposure Risk Assessment Toolbox. And the idea is to have all of these tools co-located on an Air Force accessible drive, and hopefully also other parts of the military, so that they can, that you don't have to be a coder, you don't have to be a toxicologist to quickly check your chemical out and make some informed decisions. And we do actually have a demo of that on the Air Force Google Cloud right now. And then we have a mobile app that we have developed for guiding decisions and improve situational awareness in situations where you may not have comms and you may not have a toxicologist on the scene. This I'm not going to talk about today, but just to let you know, all of the models that we're building then are meant to go into these products so that people can actually use them. This is an example of the Chemical Exposure Risk Assessment Toolbox, which lives on the Air Force Google Cloud. As I said, currently we're focused mostly on bringing together all of the exposure information or exposure limit information that we can get on any and every chemical we could find, so that we can quickly make a decision on potential risk given an exposure scenario. And I am going to try to kind of speed through some of these, and hopefully I don't go too fast, but the I mentioned that we are trying to develop models for risk. We have an in-house strong capability in QSAR machine learning based modeling. We have a publication record there focusing primarily on neurotoxicity and lung toxicity, because those are two endpoints that are particularly of interest to us. But in addition, we just recently published this publication for acute toxicity, but more focused on oxidative stress. And we used a hybrid approach using both structural alerts and message passing neural network model to identify chemicals that activate NRF2. The interest there was actually trying to delineate between oxidative stress-induced NRF2 induction and actual direct induction of NRF2, with the idea that there could be risk mitigation strategies with supplements that would reduce oxidative stress response. Oxidative stress response is not just chemical. Obviously, it's not just part of the chemical response pathway, but it's very important in fatigue, in exercise-induced fatigue, in cold stress and heat stress. All of these stresses also affect oxidative or reactive oxygen species. And just kind of interestingly pointing out we ran then these models against the drug bank repository to look against drugs that had already been approved for use. And a good chunk of the chemical of the drugs that came back as antioxidants are related to either vitamin D or vitamin A. And so we're just kind of looking into those. Vitamin D is a pretty common supplement when you're in, say, the Arctic region where it's very dark all of the time. So there's some just kind of hint that those kind of things could potentially be useful. We also have models in development for the lung, especially acute lung toxicity, kidney acute toxicity, and acute neurotoxicity. And we're also on the neurotoxicity end of things very interested in non-lethal effects like reduced cognitive performance. I want to highlight, I'm just highlighting some of the projects I think ICFAM will be interested in and ICFAM stakeholders will be interested in. But one of the big questions we have is what in the world do we do with the data poor endpoints, so we can make some really beautiful QSAR models for NRF2, because there's a lot of data on NRF2. But we actually have done a systematic review of target organ toxicity for acute toxicity, and there's not a lot out there. It's less than 15 models that we could actually use, 3 that would actually be helpful to us. So, you know, then what are we going to do? Because we need to expand out our suite of models. And the question here I've been working on with Johns Hopkins Applied Physics Lab is, can we use multitask machine modeling to improve and the generalizability of QSAR models? And so this is just a picture of the ToxCast data when you do a bi-clustering to look at kind of the distribution of the chemicals and assays that were tested in ToxCast. And so you have this kind of area of like really dense testing. And then you have a lot of areas where there's very little testing. And if you've been working with this data, I'm not telling you anything new. But then, you know, we're testing whether the solution to this might be to say, instead of having a single QSAR model for every endpoint, we make the model, we have an endpoint now. That works really great if you have a lot of data, but if you don't, what are you going to do then? The idea here is instead we have a single multitask model that starts from the beginning and is moving towards all of those endpoints simultaneously. The advantage here is that each model learns from the other models and allows us to improve our predictions. And we've done a little bit of it. This is really just a proof of concept. And I think it's like 5 out of 7 times it improved the model prediction to use the multitask learning for low density data. And this was just our kind of first go at it. So we're actually getting ready to start some more work with them, looking specifically at targets of interest for us. This is a project I'm doing with RTI International. And they published a paper a couple years ago, using as part of the acute toxicity modeling collaboration to use QSAR chemical space modeling to cluster chemicals and then use those clusters to identify which in vitro assays you should run from the ToxCast library if you wanted to know if a chemical was highly toxic or non-toxic. I wanted to put this into practice and say, okay, we have a few thousand chemicals we're interested in. Would this actually give us a plan to reduce the number of assays? So, you know, without this, and this is from their paper, but without this model, in order to really rule out acute toxicity, you need about 127 assays. This is an empirical calculation they did. But with this clustering model, you're less than 5 to get the same information. So we wanted to try it out, and that's what we're doing. We're looking at 10 different chemicals and basically running them through this clustering model. It tells us what chemicals we should test, and we're going to test them in those, plus some others that we think should light up. And this leads into another case study we're doing with the Applied Physics Lab at Johns Hopkins to really test out this entire program. And I wish I had a better slide for you, but this is what I had cleared. So the idea is to say, okay, those 10 chemicals I just told you about, let's actually run them through it. Let's run them through all the QSAR models that we have. Let's run them through all the tier 1 in vitro assays that are recommended by that model. Not all the assays in the world, but the ones that are recommended by the models. And then let's take them into organotypic models that we would expect them to the target organ to be based on the preliminary data above. And let's really see, could we have made a decision about these chemicals for a deployment situation based on this tiered testing approach that we're going to recommend to the Air Force, right? So this really puts the rubber to the road, or this is where the rubber meets the road. This is where we say, okay, you know, where is this working and where are the potential technology gaps? We're building and this I said is a collaboration with the EPA, and the more honest truth would be they really were generous and helped us. So I really appreciate this, John Momba and Caroline Ring. But the question here was, if we have the HTTK model that seems to be doing really well with really rapid assessment of chemicals in the ToxCast library. But what if the exposure is different now? Because we're not in the Air Force, you know, we're not in the business of lifetime low dose exposures, right? We want to know acute exposures. We want to know in garrison occupational exposures, 5 days a week, 8, or we want to know emergency response or even deployment, like you're going to live in this area for a year. And so we are altering the HTTK code to describe our population of interest and her exposures of interest. And then we're applying that as part of a different case study that I don't think I'm talking about right now. But this just kind of shows you that the population does differ. We obviously have a exceptionally healthy population in the Air Force, but and we're age limited, but we want to we want to make sure that we're building the models towards our population. And we actually have a collaboration starting now with NASA, where we're going to look at space flights. That's pretty exciting too. And then just the last couple, I hope I'm not, okay, the last couple things I want to talk about are more on the exposure end of things. And I'm not traditionally an exposure person, but the question we have is, can we be proactive about understanding risk before the exposure happens, right? And one of the things we're doing this again with the Applied Physics Lab is saying, can we use natural language processing, AI-assisted natural language processing to identify which chemicals are becoming popular, what's being developed, what's getting out there, and people are starting to say, hey, this is really useful, we should start putting it in products. This is you can think of this as like understanding that PFAS was going to be really popular before it became really popular and now is in everything. So it's also, you can think of it as a way of avoiding regrettable replacements. So what we want to do, we took all of the chemicals that we have in our Air Force database and we broke them down to types of chemicals, like classes of chemicals, and then made ontologies around those classes of chemicals to see just in general, what kind of chemicals are the Air Force probably going to be interested if they become developed. And then let's search public space forums for it, not just PubMed, but the internet, and identify when are the mentions for these chemicals really becoming more and more and more. And this, what we're calling citation bursts, and there are some publications on this, we didn't make up the concept, gives you an idea that, oh, wait, these are becoming popular, they're becoming useful. We should probably find out now if there's a concern for their use. And this I'm really excited about because it is showing real potential. And we're seeing things with the compounds that we looked at where you'll see kind of an initial bump in the citations where people are figuring out how to synthesize it and what it can do. And then later, so a few years later, you start to see the toxicology studies coming out, right? And that's exactly the point, because I want to bring those two together. And then finally, we can advise, we can advise acquisitions and say, no, that's not the one, right? Like, let's find a different one. A similar point is predictive chemistry for thermal degradation. So where we sometimes run into trouble is when a material is burned or there's pyrolysis going on. And now we might have done the chemical risk assessment for the parent chemical, but not only have we not done it for the mixture, but we haven't done it for the thermal degradation products, which could potentially be toxic on their own. It's impossible to burn every material in the world and figure out the thermal degradation profile, right? So we are trying to use machine learning models to improve upon current predictive chemistry models to identify likely degradation products. And then we can run those degradation products through our predictive risk models to get an idea of which of these products should we worry about. And then we can go back and be more specific to find out whether those actually are in the product or a product of degradation. So it would be a quick way to kind of triage what are our actual concerns here. And I don't know where I am, but I'm at the end of the line. Yay. Okay, so I apologize if I ran over, but and I have to say I probably don't have everybody on the slide. We have a lot of help, but over here on the left are the team that are actively working at AFRL with me, and then we have a lot of help. So thank you.
Awesome. Thank you. Stay here. I know we have at least one question online, Emily.
All right. Thank you for that, Rebecca. This question comes from Annie Jarabek at EPA ORD. She's asking, can you clarify if your lung models are only tracheobronchial or also pulmonary? Is the nasal region included?
Actually, Annie, I've been planning on trying to get in touch with you, so. So we have a lot, one of the projects I actually didn't talk about today, and it's ongoing, but we actually just got the funding that you wrote a letter of support for. So we're about to have more work along this line, but is evaluating the NAMs for lung toxicity for gases and particulates. And that is a collaboration with multiple military labs looking at in vitro models plus in silico models. And those will have components of more than one region, but not the nasal region right now. But we are trying to build an IV-IVE model for gases that includes the reactions that happen in the upper airway so that we can better understand the dosimetry in the lower lung. But we're just at the beginning of that.
I'm sure Annie has thoughts on that and would be very helpful for you to
Yeah.
talk with.
Oh, yeah, we've already noted we're actually using
Any
one of our papers.
questions in the room here? We'll move on. Okay, thanks.
Yeah, thank you.
Okay. So our next presenter, Val Adams, is online, and we are going to share your slides from this side, correct?
Correct. Thank you. So let me just go through disclaimers while you're doing that. The views expressed in this presentation are my own. I don't represent the official policy for the Uniformed Services, Department of Defense, Defense Health Agency, U.S. government, anybody else that might want to pile into that list. Anything I say that might be mention of a non-federal entity is not an endorsement of that entity or their products. And all the material presented today is also publicly available. So thanks for the opportunity to provide an update for the Defense Health Agency and the DoD on our various non-animal testing approaches. Next slide. So military exposures have multiple stressors, and the service members or warfighters are a unique population that are younger and healthier compared to the general population. Compared to the general population, service members and first responders are grouped here as well, have different performance and readiness requirements. The goal for this presentation is to provide a brief background and context for the DoD biomedical research and evaluation enterprise and the ongoing efforts to identify potential health hazards more rapidly and, where feasible, with fewer animals. Next slide. To protect the warfighter, the DoD evaluates toxicity, chemical exposures, and risk to provide a holistic force health protection program. Today, I'm going to go over the biomedical research enterprise and funding mechanisms, an example of exposure monitoring, some currently fielded non-animal methods, followed by approaches that are currently being investigated, and finally a summary. Next slide, there we go. Just as an introduction, the Defense Health Agency, or DHA, was established in October of 2013 by the DoD to support the uniform branches of the DoD and to direct the execution of joint shared services such as military treatment facilities, research and development, and public health. Additional reorganization and redefining of roles came in 2017 in the National Defense Authorization Act and subsequent congressional guidance. Since then, the DHA has been consolidating and reorganizing the various assets. In 2022, my organization, formerly the Army Public Health Center, became the Defense Centers for Public Health Aberdeen. There is also a Portsmouth branch that represents the Navy and Marine Corps and a Dayton branch that represents the Air Force. At the DCPHA Toxicology Directorate, we support both the DoD. We support the DoD through consultation, toxicology assessments, and testing. The medical research and development components, some of you might be familiar with, most of those folks out at Fort Detrick. They are currently in the process of rolling into the DHA enterprise. The Army, Navy, and Air Force maintain research components as well. You just heard from Dr. Kuhl and her work on in silico models and predictive toxicology. And in previous years, individuals from DevCom and NAMRU Dayton have presented in this public forum. I have starred the organizations that are members of the DoD Tri-Service Toxicology Consortium. I am the current chair for the TSTC, and our goal is to support the toxicology needs of the DoD and identify collaborative and cross-cutting efficiencies. And as Dr. Casey mentioned yesterday, the DoD is also engaged in collaborating with the VA to address long-term effects and exposures. The CDMRP originated in 1992 via congressional appropriation to foster novel approaches to biomedical research in response to the expressed needs of its stakeholders, which are the American public, the military, and Congress. The CDMRP invests in groundbreaking research across the full spectrum of research and development, including basic, translational, and clinical research, by strategically funding high-risk, high-reward research projects that other agencies may not be willing to fund. The CDMRP addresses critical research gaps. CDMRP funded research benefits not only military members, but military retirees and family members, and the civilian population as well. There are 38 funded programs, one of which is the Toxic Exposures Research Program, or TERP. The TERP aims to support research that will significantly impact the field of military-related toxic exposure research and or patient care, with the intent to provide solutions for service members, veterans, and or the American public who have been or could potentially be impacted by military-related toxic exposures. The TERP coordinates with similar activities in the VA and encourages collaborations between military and or VA institutions with non-military, non-VA institutions. $30 million has been appropriated to support this research. And in 2022, stakeholders identified critical issues to include a lack of data, poorly defined exposures, and emphasized a need for coordination and collaboration across federal agencies, collaborative research, and additional funding for foundational research. At the end of this presentation is a link to that document. It's got some really great information, and I would encourage everybody to go and take a look at their stakeholders meeting. Next slide. Under the Military Operational Medicine Research Program, there are environmental toxic exposures research objectives to identify and develop biomarkers of exposure, biomarkers of effect assays, to identify and improve technology for rapid screening of toxic chemicals and materials, and to provide health or toxicological data to fill data gaps for military relevant chemicals, materials, and environmental hazards. Next slide. And this is an example of a more novel approach for identifying hazards. This current proof of concept study is a collaboration between the DHA and the VA to investigate the utility of dry blood spots for quantifying analytes associated with exposure and adverse effect biomarkers. And I believe Dr. Casey might have mentioned this yesterday as well. Next slide. So currently fielded non-animal methods. Next slide, please. The Developmental Environmental Safety and Health Evaluation, or DESHE, is a tiered evaluation paradigm that pairs funding levels with testing objectives. The material acquisition life cycle that starts with the identification of a need and follows the solution from inception through the research, evaluation, development, fielding, use, and finally disposal of that item or system. Early in the process, between milestones A and B, is where many of the go, no-go decisions are made. And on the right side of this slide is a detail of the rigorous environmental health and occupational safety evaluation that occurs within the milestone A and B markers. The DHA Toxicology Directorate uses a phased approach to evaluate toxicity with the level of effort scaled to that budget activity and technology readiness level. So the TLRs are shown down at the bottom. And the TRLs will have, for early novel concepts, are going to be levels 1 through 3. Applied research and translational research and solutions for military problems occur in 4 and 6. And the prototype maturation and demonstration is in TRL 5 through 8, with production and distribution in TRL 9. These TRLs are very similar to the biomedical products line development as well. And I'm mentioning these because if you see funding that comes out, that funding is going to be directed at 1 or 2 of those TRLs. And so it's important that that people that are interested in getting funding for some of this work are appropriately targeting their strategies to those TRLs. Currently, the Tox Directorate uses computational methods, bacterial test systems for mutagenicity and acute aquatic toxicity, and in vitro systems for skin sensitization, irritation, and corrosion. And I would like to point out that one of the challenges for testing military relevant compounds is that these compounds are often not adequately represented in the computational toxicology training sets. And they do present some unique chemical structures as well. Next slide. Okay, so approaches that are currently under development. Toxic exposures to the warfighter go beyond just burn pits. And on this slide, I would like to call attention to the variety of exposures that service members may encounter. I would also like to point out that at least some of these exposure scenarios are not limited to the DoD, but would be extended to first responders and emergency response teams from local, state, and federal agencies. And so these, pose these questions are we measuring the physical performance effects? What are the cognitive effects? And is there any other sort of performance outcomes that interact with the toxicology and toxicity of any potential exposures? Next slide. So one example of work that is ongoing in mode of mechanism of action, this work is being conducted at NAMRU Dayton, and they are investigating using a variety of NAMs to more fully characterize the inflammation-based mode of action for vanadium. Next slide. For in silico approaches, you just heard the extended version of this from Dr. Kuhl. Her group is working on predictive toxicology models. And then the DCPHA Tox Director is also collaborating with her group to refine protein ligand binding screening for neurotoxicity and receptor-mediated toxicity. And additionally, the TSTC has established an internal database and work group amongst our parties for sharing computational modeling and resources. Next slide, there we go. And so at DevCom CBC, so this would be an Army group, not DHA, they're making great progress with multiple platforms to evaluate chemicals across tissues and organotypic type systems. This information was also presented, I want to say 1 or 2 years ago, so just providing a brief update that they've moved from the in development for most of these tissue types to having functional systems for them. I'd also like to point out the tissue is featured there in the middle. And from yesterday's presentation, I would expect that Dylan will be reaching out to Suzanne for some more information on that. Next slide. Yeah. Okay, so in summary, to bring it back where I started, the expectations for military personnel and first responders are different than the general population. Importantly, are we measuring these endpoints that capture subtle effects? Are we capturing the correct timing of effects? And such as delayed effects from short-term or intermittent exposures, And are we identifying nascent or emerging chemical hazards that may have delayed effects? And the DoD, Defense Health Agency, have substantial and enduring efforts to characterize these hazards to the warfighter, the workforce, and associated communities. And there is significant investment in new technologies and approaches to include biorepository sampling, wearables and sensors to capture exposure, in vitro and microphysiological systems as systemic toxicity surrogates, and then the computational approaches to predict future hazards. Next slide. And so with that, I'm going to thank Karen Mumi, who's my co-chair for the TSTC. And there are some sources listed there for the groups that I mentioned in the presentation. And I am happy to take any questions.
Great. Thank you so much, Val. Are there any questions in the room?
Thank you. That was a really interesting presentation. This is Paul Locke from Johns Hopkins, and I'm going to ask a very naive question because I don't know anything about your field. My question is, how do you evaluate, or do you evaluate the robustness of any of these techniques if they need to be field deployed?
So are you asking for like the skin sensitization, for example? Or are you asking about the sensors?
Oh, I think he was asking about toxicity testing methods and their ability to be deployed in the field. I'm getting it now.
Yeah, so I can't necessarily directly answer that. I would say that, you know, there's considerable effort on the ruggedized, you know, being able to have something that is field deployable. There are certainly some instrumentation that have been successfully deployed, not necessarily specifically for the military, but you know, there are field deployable PCR machines, for example. And you know, one of the other challenges often is, you know, temperature control, and there are some other aspects, you know, power that can make it very difficult to be doing testing out in the field. I think the preference would be to better capture, whether that's through blood, whether that's through some other filtering mechanism, to capture exposures out there, toxic, you know, chemicals out there, and then bring them back to a little bit more centralized place for evaluation.
Okay, thank you. Any questions online? Okay, thank you very much again for your talk.
Thank you.
Okay, I now have the pleasure of introducing myself to give you the NICEDM update. And I'm just going to warn you ahead of time. I have a lot of content in here and I'm going to try and stick to time, and I'm going to speak really fast. So just buckle up. Okay, so I am Nicole Kleinster. I'm the NICEDM director and the executive director of ICVAM. I love this event every year because it gives us an opportunity to highlight all the amazing work going on at all of our federal partners, but also all of the work that NICEDM does in support of ICVAM, develop and evaluate and validate new approach methodologies, and to develop tools that we hope are of broad use across the scientific community to support using NAMs in various decision-making pipelines, be those in research and development, in basic science, or in regulatory decision-making. The first thing I would love to start with is a very exciting announcement. So up until this year, ICVAM has been comprised of 17 agencies. We had NIST join us in 2017. We had the VA Office of Research and Development join us in 2020. And as of just a couple of weeks ago, we have had the National Center for Advancing Translational Sciences request official membership in ICVAM. This is a funny situation because NCATS has been participating in ICVAM essentially since its inception, and probably the only reason that they weren't already an official ICVAM agency is because they did not exist when the ICVAM Authorization Act was passed in 2000. But this has now been formalized, and I am working with Dr. Rutter to identify who who the agency representatives will be for NCATS. But as I mentioned, we already have several subject matter experts from NCATS on ICFAM work groups. So that's very exciting. And I will just take the opportunity to highlight a couple of recent collaborations that we've had between NICEDM and ICFAM agencies and NCATS. The first is a evaluation of different skin barrier models and excised human skin for dermal absorption studies in vitro. And this is really a collaboration and a study that was led by the FDA and their National Center for Toxicological Research, but NCATS and NICEDM participated in this to look at various commercially available reconstructed human epidermis models, a synthetic barrier membrane, and the excised human skin in terms of their performance. A collaboration specifically between NCATS and NICEDM was to try and sort of operationalize some of the available NAMs for skin sensitization and make them more high throughput. So they developed an ultra high throughput screening version of the keratinescence assay, screened the entire 10K compound library, used that to build a QSAR model to predict keratinescence results. And then based on some compounds prioritized in that screen, tested them in the direct peptide reactivity assay and the CD86 and CD54 surface expression assay, and ran them through some in silico models to apply the defined approaches from the OECD test guideline 497. And then the most recent one is to identify potential developmental toxicants that could be acting through CYP26A1 inhibition, which is a critical component of retinoic acid signaling pathway and its metabolism. NCATS and us and the US EPA worked with Promega to adapt their biochemical CYP26A1 inhibition assay using human recombinant enzyme and a specific substrate to the high throughput screening format, and tested it with well-known CYP26 inhibitors in a validation study that is just accepted, I think, last week into Biotechnology Journal. This is from the SOT poster. So that publication should be available very soon. Integrated Chemical Environment, or ICE. This is our platform that we use to deliver high quality curated reference data and access to computational toxicology tools in an effort to democratize these resources across the scientific community and not require that people understand how to read or write code when being able to do things like in vitro and vivo extrapolation, or run PBPK models, or do chemical-based read across, or interact with a high quality curated version of the high throughput screening data. We've had a number of different updates to ICE since the last public forum, and we have a big one coming in August. And as we referenced earlier, tomorrow we have a hands-on ICE training. So there's still a couple spaces available. I think we have about 30 people registered for doing that in person, mostly across our federal agency partners, which is exciting. And we have user stats on ICE that help kind of take the pulse of how well we're doing in terms of supporting the scientific community. Last month's Google Analytics report, we had about 1,800 users, of which 500 are return users and 1,300 were new users. So and those come from all over the world. I mentioned we have a curated version of the high throughput screening data. Part of that curation is incorporating things like analytical chemistry information on the samples that are tested, so we know whether they pass QC or not. But another big part of that curation is mapping those assay targets to different toxicological mechanisms and modes of action, such as the key characteristics of carcinogens. And we do this via multi-stakeholder working groups, including our federal partners at EPA and NIEHS, our academic partners at Berkeley and Texas A&M, and then the International Agency on Research on Carcinogens. And this is part of an artificial intelligence workflow that's being developed to try and prioritize and screen compounds for their potential carcinogenic effects. Another resource that we have supported the development of is a resource called BioBricks, and this was recently funded under a new NSF grant called the Prototypical Open Knowledge Network. NSF actually requires that their grantees identify a federal partner for identifying use cases. NICEDM is the federal partner with InSilica here. And really the problem that this is intended to solve is this realization that when data analysts want to build models or analyze data, almost half their time is spent loading and cleaning that data. And BioBricks solves that problem by doing that for you. So you can essentially load every publicly available toxicological database, including ICE, but also including things like PubChem, Chembl, CompTox dashboard, et cetera, with a one-line command. Another resource that we put a lot of time and effort into is Opera. That's our open QSAR modeling suite. Over the last several years, Kamel Mansouri, our senior computational chemist, has been busy updating many of the different Opera models. So Opera covers things like physicochemical properties, environmental fate parameters, different ADME properties like plasma protein binding or intrinsic clearance, and different toxicity endpoints, many of which you've already heard mentioned during this forum, that are the result of global collaborations to crowdsource and build consensus models for endpoints like endocrine disruption or acute systemic toxicity. Opera predictions are available through ICE, but it's also available as a standalone containerized downloadable piece of software on GitHub. And so far we've had over 7,000 downloads of Opera. And this allows folks to do things like run it behind their firewall on proprietary chemical structures or on chemical structures that are submitted under regulatory submission packages, for example. As I mentioned, Opera is available via ICE. So we've taken over 1 million structures and run those through Opera so that you can access those predictions via the ICE dashboard. But it's also available through the EPA Comtox Chemicals dashboard, via the FDA's Precision FDA platform, and via the OECD QSAR toolbox. So really trying to harmonize with both our federal and our international regulatory partners to increase accessibility of these computational tools. And one really nice example that just came across my desk last week is a very recently published paper, and we're increasingly seeing more and more of these publications in the literature where groups completely separate from NICEDM or ICVAM are using our tools in their workflows. So this is a workflow where they have derived human equivalent points of departure exposures for almost 2,000 organic chemicals, and their workflow depends very heavily on properties predicted from Opera that were used as features in a machine learning model, and exposure data from the US EPA ExpoCast program that's available through ICE to facilitate comparisons and derivations of margins of exposure. And this is a really top-notch group with participants from all over the world. Another nice part of Opera is a piece of the Opera workflow is actually the ability to standardize chemical structures to make them what are known as QSAR ready. So desalting them and normalizing them, removing duplicates, et cetera. And that QSAR standardization workflow has just been published in the Journal of Cheminformatics as a free and open source workflow that supports QSAR modeling, not just in Opera, but for groups worldwide. And it's available for download as a KNIME workflow. We also, one of the toxicity prediction models in Opera is to predict acute oral systemic toxicity. So the Collaborative Acute Toxicity Modeling Suite, or CATMOS, was this global consortium where we crowdsourced QSAR models to predict the oral LD50 and then combined those models using an ensemble approach that leverages the strengths and compensates for the weaknesses of any individual approach and provides these incredibly comprehensive, robust model predictions that allow you to predict the acute oral toxicity for any chemical in the known chemical universe with a defined structure. And so then we worked with our regulatory colleagues to do an analysis of how well CATMOS performs when predicting the LD50 of technical grade active ingredients for pesticide products. And we found very high categorical concordance for essentially non-toxic chemicals with an LD50 above 500. And, you know, the sort of take home here is that predictions of the non-toxic predictions were found to align extremely well. And we're hoping that this will be the basis of a future waiver guidance from the US EPA, where if a chemical is predicted to be non-toxic, so an LD50 say above 2,000, then that waives the requirement, or that's one line of evidence to waive the requirement for the animal test. We're now looking at doing the same thing for acute inhalation toxicity. So we are compiling a database of LC50 data. You can see that we have a number of different data sources that we're using largely from our federal partners, both in the US and in the EU. That data has gone through a really extensive and arduous cleaning and curation process, where we've flagged data for quality issues. We've had to try and convert them all to the same types of units, account for different phases of exposure like aerosol or vapor, et cetera. At the end of the day, we have a database. It's not as big as the one we had for CATMOS, where we had almost 12,000 substances. Here we only have about 1,000 unique chemicals. About 760 of those are going to be used to support a collaborative modeling effort. And if you're really eager to go play with the data, you can just go ahead and download it from ICE. And then another really outstanding effort that we have ongoing is to try and, again, democratize cheminformatics methods among non-experts and facilitate their use across the scientific community. This is a pipeline known as MOVIS, the Modeling and Visualization Pipeline. Again, we try and use open access tools wherever possible. So this is developed using the KNIME platform, and it has things like data access and storage, data mining and curation, analysis and visualization, modeling and prediction. And all of these tools together have a graphical user interface with really extensive documentation to help sort of non-expert users make informed choices about what to do at every stage of a pipeline like this. Sort of the first application developed in this MOVIS pipeline was a chemical grouping workflow. So this approach of categorizing chemicals with common characteristics, and that could be phys-chem properties, it could be toxicity values, et cetera. And this has a lot of different applications, potential, assess chemical diversity of a library or select representative compounds for testing, look at different mechanisms of action, support chemical risk assessment, et cetera. Another area where we're trying to leverage computational tools to help make our lives easier is in automation of study data curation. So any of the data that you access via ICE is high quality. Most of it is highly manually curated, which is an extremely arduous and time-consuming and resource-intensive process. And so wherever we can, while still maintaining the human in the loop, we are trying to use augmented intelligence approaches to automate different pieces of that pipeline, from identification of high quality studies to extracting information from those studies, both methods and study protocol details, but also effects and outcomes. And then taking that extracted information and actually programmatically using controlled vocabularies and knowledge organization systems to apply those standardized terminologies to the extracted study information so that it can be joined with other data sets that use those same controlled vocabularies and create supersets of data that can be used for a variety of different applications, building predictive models, validating new methods, assessing the totality of evidence for chemical effects, et cetera. This piece, the annotation piece, we're probably furthest along. We just published a paper in EHP last month describing code that we built to programmatically annotate extracted information from prenatal developmental toxicity studies that have been run over the last several decades at the National Toxicology Program and were submitted to the European Chemicals Agency. But we are making lots of progress on the sort of proximal end of that pipeline as well. Large language models have been a game changer for a lot of different things, but in particular, the field of natural language processing, I think, has experienced an absolutely exponential jump in productivity based on large language models. So we're developing right now a pipeline to identify high quality publications, extract information from the studies, and sort of assess the adherence of those studies to minimum criteria that we have identified from things like regulatory guideline protocols to assess whether published studies in the literature would be considered high quality enough to be included in regulatory decision making. We have another really, really useful computational tool called the Defined Approach for Skin Sensitization app, the DAS app. Kim Toad from the Innovative Nicetrum Group has been leading the development of this, and this was just published in BMC Bioinformatics earlier this year. This is really to operationalize the defined approaches that you see in OECD Test Guideline 497. So these are a number of rule-based integrated strategies that combine in vitro, in chemico, and in silico information sources to predict an outcome of skin sensitization, whether that's a hazard outcome or a potency categorization outcome. And to remove the potential for human error, we have created a sort of to minimize the potential for human error, we have created a user-friendly application with a downloadable template where folks can upload data and with a few clicks of a button, they can run the different defined approaches and get performance statistics. And then we're working on some major UI updates to accommodate some new features here to include things like borderline results in the assays, doing some data visualizations to compare with reference data, look at the influence of physicochemical properties that can have a big impact on the suitability of different in vitro test systems, et cetera. Continuing with the skin sensitization theme, we, as part of the OECD Defined Approach project, we curated the largest ever reference database of human clinical reference data for this endpoint. So human predictive patch test data is really, I think, the gold standard for comparing new approaches for skin sensitization. But up until this project, there wasn't really a curated database all in one place. And so that database has now been published at the end of last year. And then based on that data, we have, with our colleagues at the German Institute for Risk Assessment, the BFR, we have proposed a novel classification approach, so updates to the globally harmonized system for classification. And that was just published earlier this year. And we have, of course, an app under development to try and apply that new classification system. And then to move from just hazard and potency classification to true quantitative risk assessment for skin sensitization, we've been working with our colleagues at Unilever to develop out and adapt the skin allergy risk assessment model. That's a model that they originally developed. It's a Bayesian statistical model. So it's a probabilistic approach to deriving a quantitative point of departure for risk assessment that accounts for variability in that input data and gives you, uncertainty quantification. They originally developed it based on about 85 or so compounds commonly found in personal care products. We dramatically expanded it with several hundred more compounds from the ICE database. And this is currently under evaluation by the OECD expert group for potential inclusion into test guideline 497 as the first defined approach that provides you with a true quantitative point of departure for use in risk assessment. And again, we have an app for that. You're sensing a theme here, right? So we have hired a software developer to develop a fully containerized version of the Sarah ICE model that makes it easy for people to apply, something which is, otherwise I think a pretty challenging complex statistical modeling approach. And so again, you have a template, you upload data, you can run the model. One of the advantages of a Bayesian model is that it can handle subsets of information sources. So you don't have to have every in vitro assay under the sun to run the model. You can run it with as few as 1 or 2 in vitro assays. You just get much wider confidence intervals around the dose that gets predicted. And these are some screenshots from the newly available model. It's not published yet and it's not publicly available yet. However, if you are interested in beta testing it, email Emily. And she will share it with you. So finishing up our skin sensitization topic, you heard Charles talk yesterday about the ESA. It was accepted just last month onto the OECD work plan for inclusion as a Me-Too assay in TG442C, which assesses the first molecular initiating event of the skin sensitization AOP. This was an ICVAM validation study, so you can see the labs are largely ICVAM agency labs, and Burleson Research Technologies is actually the immunotox testing contractor for NIEHS, for the Division of Translational Toxicology. And this is a really good example of taking an existing assay and making it much more accessible in terms of the probe that's used is a fluorescent or colorimetric probe. It doesn't require really specialized machinery or equipment. It's much more affordable and easier to run. And the peer review of this validation study is underway. Okay, switching gears just a little bit, eye irritation. We had a collaboration with the PETA Science Consortium International and the EPA Office of Pesticide Programs to assess the applicability of different non-animal methods to agrochemical formulations and develop defined approaches that could leverage the strengths and the human mechanistic relevance of these methods to predict the full spectrum of eye irritancy potential. This is really a very good example of a real nuts and bolts application of the concepts and the scientific confidence framework articulated in the ICVAM validation workgroup report. And this was just published also earlier this year. So the defined approaches that predict the different EPA categories. And we now have a manuscript in preparation to try and sort of globalize this and predict the different GHS classifications using other defined approaches that share quite a bit of similarity with the EPA classification defined approaches. We're also constantly working with our collaborators at the US EPA to further develop and build out the models that are being used for in vitro to in vivo extrapolation. So PBPK models are, essentially a mathematical representation of the body and how your body breaks down and distributes and excretes or not different chemical exposures, be they pharmaceuticals or environmental chemicals. And I steal this from John Wambaugh. I like this description. They range from very simple, just your body is a bag of water and everything just equilibrates immediately, to much more complex. So, systems of partial differential equations that mimic each of the different tissue compartments in your body. One of the areas that is sort of lacking in some existing sort of high throughput toxicokinetic models that can be applied across the chemical universe is explicit representation of the blood-brain barrier. So we're working closely with John and his colleagues to update the HTTK models to bring in the blood-brain barrier either via a diffusion limited brain compartment, considering that permeability, or just a perfusion limited model. And we're also looking at bringing in adipose tissue compartments as well, partially because that's, I think, a really high priority for environmental chemicals, many of which are highly lipophilic and could potentially be bioaccumulative in the body due to their affinity for adipose tissue. We've also been looking at a comparison of various PBPK models and their applicability to developmental neurotoxicity. So evaluating both commercially available models and more open access available models like the high throughput toxicokinetics platform from the US EPA. And this work is currently being written up for publication. We have a very strong program in developmental neurotoxicity, partially due to the addition of Dr. Hogberg to our staff 2 years ago. And we've been working with international consortium to help identify reference chemicals, as was discussed earlier, but also to identify what can be sort of characterized as training set chemicals. So there was this battery of in vitro assays that was included in an OECD guidance document in 2023. And there are sort of a minimal set of chemicals that's needed for assay transferability to demonstrate robustness and reproducibility as a key part of the validation process. And so we're working on developing those recommendations and providing those lists to OECD this month. We didn't have I think last year we had a full presentation about the DNT HEI program, but we were already borderline making this the NIEHS show. I think we had like 4 or 5 presenters from NIEHS. So just decided to dedicate one slide to this incredibly important program that we are supporting through the Division of Translational Toxicology, where we're supporting screening efforts of chemicals that have been nominated by our stakeholders. So many of our agency partners have nominated compounds of concern, and those range from things like PFAS and flame retardants and industrial chemicals to things like recreational drugs and cannabinoids and botanicals and food additives. And so the first phase, 115 chemicals, that testing has been finalized. The manuscript is in prep. The second phase, another 108 chemicals, including complex mixtures, that testing is ongoing and will hopefully be completed later this summer. And then phase 3 is another 120 chemicals, and that selection process is ongoing. And then we recently had a Scholar Connect student from NC State that Helena supervised, that did a systematic review of the literature. So following the protocol for both systematic review questions and bias analysis that EFSA, the European Food Safety Authority, has developed to look at DNT IATA case studies and identify potential reference compounds that can be tested in these DNT assays. And then also looking at the existing data that we have for those compounds to try and help identify molecular initiating events and provide more insight into not only the mechanisms of their potential toxicity, but also whether or not there is concern based on exposure for mothers and for fetuses. And we use ICE to do that sort of IVIVE and that overlap. Another area in the neurotox space that we've been doing as a collaboration with Neurocrine Biosciences, Syngenta, and Collaborations Pharmaceuticals is to identify seizure liability. So this is really more relevant to the pharmaceutical space. So it's a major factor contributing to attrition in drug development. And there's also obviously the potential for seizure-inducing effects from neurotoxic compounds in the environment. So various insecticides have been implicated for potential seizurogenic effects. And so using an AOP network and that was developed based on multiple different AOPs that already exist in the AOP wiki. This allowed us to prioritize 25 different seizure-related target families, map those to in vitro assays, both from ICE and then from Kemble, and then look at identifying reference chemicals. So almost 200 positive reference chemicals and about 35 negatives were identified from the literature, and looked for sort of commonalities and patterns in their in vitro activity against these seizure-related molecular targets. And this manuscript is also in preparation. Kind of along the theme of trying to take in vitro assays and provide better biological context and map them to molecular initiating events or key events. There's an international collaboration that includes some of our ICADM partners, so the European Commission, Joint Research Center, also Environment and Climate Change Canada, RTI International, US EPA, called the Methods to AOP Consortium. So really and this was presented at several recent meetings, including the SOT and the latest World Congress on Alternatives in August of last year. Really the overall goal is to map assays to MIEs and key events in AOPs to sort of build on and expand out the AOP infrastructure, increase confidence in AOPs, maybe serve as a foundation for developing things like integrated approaches to testing and assessment or defined approaches, and to engage method developers to sort of actively participate in that process. We had a brief update on this last year, and I'll just give you a status on where we are. So we are supporting a validation study of a human thyroid microtissue assay. The thyroid signaling is obviously a very high priority for the US EPA endocrine disruptor screening program and for many other agencies worldwide. This assay consists of primary human thyroid cells. They are commercially available or will be soon from LifeNet Health. This assay was developed in Chad Diesenroth's lab in the US EPA. It assesses thyroid hormone inhibition, and they've miniaturized it to a 96-well format, so it has really, good throughput. And NICEDM is running the validation management team, and the assay is being transferred from the lead lab at the EPA to 3 other labs, and that is ongoing. So the first initial transfer phase is complete, and then the secondary transfer phase is underway, and then the validation study will start this summer. And then another health effects innovation program that we have ongoing that NICEDM has been very heavily engaged in at the Division of Translational Toxicology is the cardiovascular HEI. Which y'all don't know from the morning is Shagun Krishna, who gave the public comment for PCRM up until a month ago, was my postdoc. And so she was with me for 4 years, and she led so much of the work in this cardiovascular space in terms of mapping different in vitro assay endpoints to mechanistic targets that are critical for cardiovascular disease onset and progression, to help us really understand how environmental chemicals might be contributing to cardiovascular disease. And she's done this in a really, really comprehensive way, not just via identification of in vitro bioactivity profiles, but also incorporating IVIVE to derive margins of exposure and bioactivity exposure relationships, but then also partnering with some of our investigators that specialize in geospatial risk mapping to look at exposure data that we have from things like the National Air Toxics database to create risk maps of the continental US where there might be an increase in risk of cardiovascular disease based on exposure to these compounds that are preferentially and potently targeting cardiovascular molecular signaling. And then she's also been leading an effort in collaboration with our integrative health assessment branch on doing a systematic evidence map of the literature that's out there that links exogenous exposures to cardiovascular risk. And that's, to really try and to gather these different lines of evidence, in vitro mechanistic, in vivo animal data, and human epi data, to give us an interactive way to assess the totality of the evidence and prioritize chemicals for additional testing. I'm almost done. We had also a really, really brilliant PhD student who visited us from Brazil for a 6-month internship. And he's from this southern region called Minas Gerais. It's over 90% of their economy is based on coffee plantations, and rural workers and nearby residents, mostly their spouses, are highly exposed to triazole fungicides. And so he had a human data set that was, in my world, unprecedented. So he had these agricultural farm workers, he had human exposure measurements in both plasma and urine, but then he had a sort of case control group from the same region, but that lived in the urban area. So he had farmers, their spouses, and then this urban group. And he looked not only at exposure, but also at things like oxidative stress markers, genotoxicity, liver enzymes, steroid hormone, and then did some risk calculations, found that the hazard quotients for the agricultural communities was much higher than acceptable, but then came and did a 6-month internship with us to understand how to apply computational workflows to this data. We have high throughput screening data from ToxCast and Tox21 on all of these compounds in our database. And so he did the mapping to the different mechanistic targets for all these fungicides, did the IVIVE, looked at the overlap, and it's like we scripted it. Not only are the targets the same, so we're seeing perturbations of steroid hormone regulation, of liver enzymes, of genotoxicity and oxidative stress biomarkers at the exact concentrations that correlate to the exposures that he's seeing in his agriculturally exposed, occupationally exposed farm workers. And not only that, the exposures that he measured that are, exactly overlapping with the exposures that we would predict from our in vitro assays, those exposures are over an order of magnitude higher than the 95th% confidence interval from the US EPA's exposure predictions, which take into account occupational exposure. And what this means, and he knows this because he also did a survey about personal protective equipment use, is that the PPE is not being used effectively, if at all, and that probably far more of the products are being used in the fields than is recommended. And so he won, by the way, 3 different awards for this poster at the SOT annual meeting, and it was his first SOT, and so it was just really exciting for everybody. But this data and this story and this publication, which we just submitted to EHP, I think is a really, really nice example of a true gold standard apples to apples comparison, human in vitro with human in vivo, but also a compendium of evidence that supports risk mitigation interventions. So it supports going to public health authorities in this region and saying, hey, you need to, intervene. You need to put funding and resources towards protecting your population and helping to educate them about the importance of personal protective equipment. Okay, the last couple slides I have are about workshops that we've run in the last year or two. A big area of focus for us, and overlap with the complementary program, is the necessity and the importance of using NAMs to address variability and susceptibility across populations. And I think that there's a tremendous opportunity here for getting genetically diverse models and representing things like comorbidities or co-exposures or different life stage susceptibilities. We had a workshop, an initial meeting about this at the end of 2022. That workshop report is finally in a final draft stage and will be submitted to a special issue of Human Genomics shortly. And I'd really like to call your attention to the QR code on the screen because this is a special issue that is being guest edited by Helena and Kim from the NICEDM group, and it's really focused around NAMs for addressing population variability and susceptibility, and we are welcoming submissions right now. Technically, the submission deadline is the end of June. Technically. Okay. Another workshop that folks from NICEDM helped organize with lots of co-organization across collaborators in the DTT was on clustering and classification of compounds. So trying to sort of evolve best practices in defining concept similarity, both in a supervised and an unsupervised fashion, basically trying to get the field to evolve past a simple like Tanimoto score based on one set of structural descriptors is how I'm going to define compound similarity. Instead, moving towards a much more nuanced and more complicated approach to defining chemical groupings. So thinking about the endpoint of concern, thinking about bioactivity as potential descriptors, thinking about physicochemical properties, thinking about varying libraries of structural descriptors, all to inform better and more evolved approaches to clustering and classification. This workshop report has been submitted to EHP and it should be published soon. It's under revision right now. And then Susie mentioned this briefly yesterday, but we co-organized a workshop on NAMs for the GI tract, looking at direct toxicity, de-risking chemicals for systemic toxicity, models for absorption and distribution, and models for allergenicity. This workshop report is also in final draft stage and will be submitted to Altex soon. Okay. I really have to take a moment and say that the group that I am privileged to lead is one of the most outstanding group of individuals that I have ever had the pleasure of working with. Most of the folks pictured there they work for a company called Innotive. So they are federal contractors that provide absolutely unparalleled and outstanding support to the NICEDM group. And we're just so fortunate to have them. And if I thought you would have the patience with me, I would call out every single one of them by name and thank them each individually. But we probably don't have the time for that, unfortunately, but they're just amazing. And none of this work will be possible without them. And then in the front row there, you see some of our federal employees. Dory Germelek is in the systems toxicology branch at DTT and is 50% of her time with NICEDM. Helena, Kamel, Pei Lee, our contract office representative, are also just absolutely instrumental parts of our group. And then of course, we have a huge list of collaborators across our ICVAM partners, but also across our stakeholder organizations in academia, in industry, in NGOs. And then just one thing I want to mention is that Kathy Sprinkle has been hard at work on our new biennial report. Part of our congressional mandate is that every 2 years we have to publish a report on all of the activities, not just that NICEDM is doing, but all the ICVAM agencies are doing. It's an incredible resource and it's web-based. So you don't have to download the whole like 300-page PDF. You can actually, and it's tagged with metadata. So you have the ability to search for like, what's FDA doing in developmental toxicity and go straight to the articles. So that's it. Thank you so much. I'll welcome any questions.
I'm a mess over here.
Well, you're presenting next, so.
I know, this is like, I don't know how I'm going to follow that. So first question, can you share the name of the intern from Brazil doing the work on pesticide exposures? I missed it because you were talking too quickly.
Okay. Yeah. I love I love a compliment like that, right? Luis Paulo de Marciano Aguilar. His picture is the only one that's not in the group picture. He's amazing and we'd love to get him back to do a postdoc.
All right, this next one is from Elaine Merrill at the Navy Marine Corps Public Health Center. Are there standardized approaches for applying QSAR and ML models? For example, are 3D MDs generally preferred over TUD? I'm wondering if this popped in. No, for example, are 3D MDs generally preferred over TUD? Is there an approach to avoid overfitting?
There were a lot of acronyms in there and I thought that I was fluent in acronyms. But
QSAR and multi-language models. 3D, I'm guessing.
Oh, 3D molecular
Molecular
descriptors instead of 2D molecular descriptors. Yeah, so it's a fair question, right? Because most QSAR models are built using two-dimensional molecular descriptors, although we do have some work ongoing to look at the utility of 3D descriptors. One of the resources that I didn't get a chance to highlight is a resource called ChemMaps, which is like basically a map of the chemical universe that's informed by three-dimensional descriptors. So it can support looking at clusters of compounds based on that. Okay. All right, wonderful. Thank you so much. And I'm now going to turn it over to Emily Renke to give us an update on the progress of various ICFAM work groups. And so I will set up your slides for you, Emily. There we go.
All right, so thank you.
Last but not least.
I know you all are really tired. So this is actually pretty quick. We should be okay. Hi, my name is Emily Renke. I am a contractor supporting NICEDM, and normally Dave gives this update, but Dave's not here anymore. So I guess it's me now. Not Dave, Maddie. Sorry, we're getting some commentary from our Air Force colleagues in the back of the room. Anyway, so just giving our update on the different work groups that we have at ICFAM, and we thought it was appropriate to put this at the end of the day. And thank you everyone for sticking around, because this is the ICFAM public forum. And we do want to make sure that we're highlighting the efforts of the work groups, as well as all of the individual federal agencies that are helping with this. So these are, as of the end of 2023, all of the work groups that we had in existence. So we had the acute toxicity, the consideration of alternative methods, ecotox, in vitro to in vivo extrapolation, validation, and a PFAS work group. It is typically preferred that a work group goes as a scope and charge, fulfills that scope and charge, and is then sunsetted into what we call an expert group. So as each of those groups fulfills their what they need to do, that is what they will do eventually. I did not realize this was okay. So the acute toxicity testing work group, this is probably one of the longer living work groups out there. It has completed its scope and charge around oral and dermal systemic toxicity testing. We've spoken about this work group over many, many years. So we've looked at, the usefulness of acute oral LD50 data for classifying dermal systemic hazard of potential toxicants. And that was completed, and there is now a waiver in place for that from EPA. We had the CATMOS project that Nicole talked about in the last presentation, so I'm not going to belabor that point too much. There has been a GHS additivity formula evaluation for acute systemic toxicity tests. And then there was a US scoping document published on regulatory requirements. Ongoing, again, Nicole touched on this briefly, the LC50 database has been finalized for doing inhalation toxicity, and we have that ongoing collaborative modeling project. Wow, these slides, I don't know what happened with these. The ongoing acute inhalation modeling project, which again, it's that collaborative effort to build a modeling approach that can predict acute inhalation toxicity. We're taking this in a slightly different direction than how we did CATMOS. Instead of putting out, these are the endpoints that we're interested in, here's our training set, go ahead and build us a model and we'll evaluate it. We're actually kind of turning that on its head, and we're bringing the modelers together first to help us decide what the best way is to approach modeling with this data set. Again, it's not large, it's not tiny, but it's not the biggest data set. And there are additional complexities around inhalation endpoints that we need to take into account. So again, we are bringing together a group in order to do the, how do we want to do it? And then they'll go off and do it individually. The consideration of alternative methods work group, the scope is to work with stakeholders to develop a catalog of incentives that could be used to encourage proposals for NAMs in conjunction with existing in vivo test methods. So how do we get more NAMs being used in conjunction with those animal tests that are that have to continue going on at this point? So they have been working with stakeholders to do interviews with them to find out what are some of the barriers and what are the, what things need to be put in place in order to further encourage this use and consideration of alternative methods. So stakeholder discussions were held between May of 2022 and May of 2023. And we had ag chem, industrial chemical, consumer products, pharmaceutical companies, academic researchers, and academic IACUC members as well. And again, it was to kind of get their experience around tox testing and NAMs and alternative. There is a white paper under development right now that will be submitted to Altex. And it catalogs the different stakeholder perspectives and covers how alternatives to traditional animal tests are considered within the development of respective organizations' tox testing programs. The ecotoxicity testing work group is also still working its way through another publication. That is looking at identifying one or new NAMs that can potentially be used to reduce, refine, or replace the acute fish toxicity test. So they're characterizing the identified methods, determining criteria that are important to the regulatory agencies in those methods. And they are collecting agency input on these various questions that are on here. And there's reference literature being brought in and updated in order to look at a kind of a comparison between the FET and the cell line, the RT-GILT W1 cell line assay. As I mentioned earlier, when a work group has completed its scope and charge, we will tend to convert that into an expert group, and the IVIVE work group did a fantastic job putting together some additional scoping papers. And this one that was published in 2022 was recognized by ATSTR as an outstanding publication, and was also recognized by Toxics as in their annual recommended review, and actually was featured on the cover of that month's journal's publication. The issue. Now that they have completed their charge, they have been converted into an expert group. So we're keeping all of those experts on a list, and we have access to them as necessary. The validation work group. The great thing about this one is that we actually published the validation, qualification, and regulatory acceptance of new approach methods. This was published in March of 2024. Nicole is presenting on it on Thursday in a neuro seminar, and we sent out the link for that earlier today. This was a multi-year effort to put this updated guidance together, and so if you're interested, go ahead and read it or listen to Nicole's talk on Thursday. It'll be a really good way to kind of get a good overview of it. Following on from that, we are working on doing a method developers forum. And this is a proactive effort to highlight and implement the recommendations detailed within the BWG report and provide opportunities for NAMS developers to interact with stakeholders around regulatory issues. Currently, we're anticipating holding 2 MDFs every year, and each iteration will focus on a specific endpoint or toxicity. The first MDF will focus on carcinogenicity. I should update that to say it's actually August of 2024. We have ICFAM agency and industry stakeholders summarizing their information needs around carcinogenicity and the potential context for use for NAMs. So participating federal agencies are listed here. We've got CPSC, EPA, OPP, OPPT, FDA's Centers for Food Safety and Applied Nutrition, and CEDAR. The National Cancer Institute, NIOSH, and OSHA are all providing presentations. We also have Syngenta providing an overview from the agrochemicals perspective, and we are still working on getting a few pharmaceutical stakeholders to present. These are pre-recorded presentations that will be hosted on the NICEDM website, and will allow for any method developers that are interested in participation in the full event that will be in August to watch those and get an idea of how their NAMs need to fit into the regulatory needs. Future topics will include developmental toxicity, reproductive toxicity, cardiovascular, neurotox, and systemic tox. Basically, we're going to try and run the whole gamut as much as we can. So if you are interested in that, you can reach out to any of us and we can provide more information. And last but not least, we have the PFAS testing and assessment work group. Current efforts on that are to put together a white paper that is on the state of the science on NAMs for PFAS. And this will include a review of available information for PFAS testing using NAMs and the overarching challenges and data gaps for application of NAMs within a regulatory assessment. Once that white paper is completed, which should hopefully be later this year, There will be a workshop or a conference session on the application of NAMs for specific PFAS. And then reporting out from that, a publication on future directions. And with that, we will take any questions. So, and then from Adam Bettman, we have, is the acute inhalation data set similar to that of CATMOS, where more data for lower toxicity or non-toxic chemicals are readily available? And do you expect it to be similar in doing a better job of making non-toxic predictions, or is it too early to tell? The data are skewed towards the non-toxic. So like with the CATMOS database, we're looking at more chemicals that are in the less toxic space. So while I don't want to make any predictions on model performance, since we don't even have a model yet, you're probably not outside the realm of guessing that it might be better at predicting non-toxics than toxics, just based on what the database is made up of. So. All right.
Fantastic. Thank you so much, Emily. Okay, so we have come to the end of a wonderful 2 days, day and a half of our 2024 ICFAM Public Forum. I think that I'm seeing lots of applause online, virtual applause. That's great. Thank you so much. I would also like to add my kudos to all of the presenters and all of the excellent questions that were asked and the discussants. It's really heartening to see the amazing progress that we can display every single year across all of our agency partners and ICFAM as a whole. So thank you all and safe travels back to wherever you may be going, be it near or far. And thank you for our participants. We had over 100 participants online for the vast majority of the time. I think we had about 160 yesterday. So really great, robust participation. Thanks, everyone.